Questions? You had a problem, I got an email from someone that had a problem submitting their online exam. Anyone else have a problem? Oh. Okay. You know, I think you could submit it until, did anybody look at the time? Till when? Till 11? Okay, so past class, okay. I thought maybe I'd had it set at, 10:20 exactly, and someone had started and didn't submit till after, okay. Okay, no questions. After class today, I'll be around here for questions. And if you can't make it, just, email in a question. And I can either I'll just do a short presentation, is the easiest thing. Okay, so today we're going to talk about metabolism. And I wanted to start with this, which we talked about last time. And this is a fairly new discovery, as in new in like the last less than 20 years, that glucose can be produced in the chloroplast, and glucose can be translocated through the chloroplast membrane. Now, glucose is not going to just stay as a neutral glucose molecule. The same thing for fructose. Is as soon as these are carried through, they're going to be phosphorylated. And one of the many reasons that sucrose is transported from cell to cell, or from tissue to tissue, let's say, is that sucrose, which is composed of glucose and fructose, when the bond, the covalent bond is formed between fructose and glucose, those reactive sites are covered up. So sucrose is a fairly stable molecule. Sucrose can be broken down, of course, by invertase, and it can be broken down by a number of enzymes, but sucrose is pretty stable. And then also, for every one sucrose molecule that's transported, that's 12 carbons instead of, in the case of glucose or fructose, which of course each are 6. So triose is going to exit the chloroplast, and triose then, a lot of things can happen to the triose. And one thing we'll talk about today is it can go into glycolysis. So glycolysis, you're all familiar with, and you've had it before. And what glycolysis is sort of the priming of the pump to produce a lot of energy. And you usually see that glycolysis usually starts at glucose. But in plants, you'll see glucose as the starting point of glycolysis. But in plants, a lot of times you'll just see triose. Why? Where's the triose coming from? I just told you where the triose. It's coming from the chloroplast. So in leaves, it doesn't really make much sense to produce a triose, then to synthesize a glucose, and then to break that glucose down to triose. So in leaves, there is glucose around, but this triose is coming here, okay? So then we want to go over these steps, because these steps, and you don't need to know the individual steps, but you should understand how energy is produced. And the first thing that happens is that the triose phosphate enters the pathway, and it can be converted to dihydroxyacetone phosphate. And if it's glyceraldehyde 3-phosphate, when this reaction occurs, NADH is made. Okay, that's important. You're going to see how important that is. So for this reaction to occur, NAD is required to be reduced to NADH. Then we go through some other reactions, and we go through two reactions, this reaction and then the reaction down here, where ATP is made. Okay? And we'll talk about this a bit more, but this is substrate level phosphorylation. Substrate level phosphorylation. The ATP is made directly in the cytoplasm where glycolysis occurs, and ADP is required for this reaction to occur. Down here, this same reaction also requires ADP. Okay? So you have an ATP and an ATP and an NADH. These two ATPs are produced in the cytoplasm. And this is different than the ATP production that we've talked about in the past, which has been through ATPase. Okay? And that was generated, those ATPs are generated by electron transport either in the chloroplast or we're going to talk about today in the mitochondria. Here, this ATP is made right here. And that's important, as you're going to see today, is because this is all of the reactions that can occur in a plant without oxygen. Okay? And I just talked about this, and I'll just go past that. And to the net is 2. So there's 2 ATPs produced. Now, keep this in mind that this is what can happen in the cytoplasm without oxygen. And we're going to talk about another pathway that also occurs in the cytoplasm. And this is called the oxidative pentose phosphate pathway. And what you should know about this is it produces NADPH. And it also produces a CO2. So this pathway produces CO2 and NADPH. And when this CO2 is cleaved off, then you have 5 carbon compounds that are produced. And you don't need to know these individual, you should know that this is where most of the ribose that's used for what in the cell? Nucleic acids. So the 5 carbon sugars that are part of DNA and RNA come out of this pathway. And then CO2 is produced and NADPH. Produces the pentose phosphates, and also it produces intermediates for lignin. If you remember, we talked about lignin earlier. What you should know is that it produces the carbon for nucleic acids. Now, I mentioned if we go back here, that this reaction, these are the reactions that occur that do not require oxygen. Okay? There's no place in here where you see oxygen required. For plants, plants have evolved a system to basically get by without oxygen, not to thrive, but just to get by. And what the system is fermentation. And we're going to talk about fermentation, then we're going to talk about electron transport in the mitochondria, and we'll come back to fermentation because it makes more sense then. Basically, there are 2 enzymes, lactate dehydrogenase and alcohol dehydrogenase. And the purpose of fermentation is not to produce alcohol, but it's to recycle NADH. Okay, why is that important? Let's go back up here. I told you that when this reaction occurs, NAD is needed to be part of that reaction. It has to be reduced or this reaction cannot occur. So if all the NAD is converted to NADH, this reaction cannot occur, which means this reaction cannot occur, this one, or this one, and no ATP would be produced. In conditions where oxygen is limited, this is all the ATP the cell has to work with. I'll tell you now that aerobically, plants, animals generate 36, 38, 40 ATPs per glucose molecule. Anaerobically, plants produce 2 ATPs per a glucose molecule. Okay? So let me flip back down here. And we have these enzymes, and here's what happens. Aerobically, pyruvate, the product of glycolysis with oxygen, is going to go through mitochondrial respiration, which we're going to talk about in a few minutes. And aerobically, pyruvate, it can either be converted to lactate, or pyruvate is converted to acetaldehyde and produces the CO2. And the reason for this reaction to occur is NADH is then cycled back to NAD, NADH is then cycled back to NAD, and then that allows this NAD then can migrate over here and keep glycolysis running. So what do we produce if we have low oxygen in our muscle tissue? Lactic acid. This is the reaction that we do right here. And plants do this too. In fact, for about the first 10 minutes, plants produce lactic acid, which lowers the pH, which then activates this pathway. Why don't we do this pathway? Why do we produce lactic acid? And nefarious, lactic acid is nefarious. We then remetabolize the lactic acid. In plants, the ethanol is just like water and goes right through the membranes. So we lose that 2 carbons. We can't recover those 2. Plants can't recover those 2 carbons. So why don't we do this? Why don't we produce a lot of ethanol? Think about an organism that would try to evolve to run away from predators, that every time you started running, you got really drunk. You know, so, you know, there's nothing that really evolved to do this other than things that can't move. Okay? Every time you water a plant, you're producing a tremendous amount of ethanol. Every time you water a plant, you're flooding the plant. And it's fine. And we know plants can be flooded for days. They're not going to grow. And if they are continuously flooded, they will eventually die. But, you know, they can get by. They can maintain cell integrity. There's other ways they can produce some limited amount of energy, but for the most part, these ATPs that are produced here, that's it. So the plant can maintain cell integrity. It's not going to grow a lot, and it's going to be able to survive. Other things we talked about, I think in one of the first lectures, how plants can actually break down part of their roots to produce pipes, openings, that oxygen can flow down to the root, to the tip of the root. Okay? Questions about this? Now, we'll talk about the other more common fate of pyruvate, and that is that pyruvate is transferred and completely burned to CO2 in the mitochondria. Okay, now what you need to know about this pathway. Well, first of all, it's a cycle. So we're going to end up with what we started with. And in the case of plants and animals, pyruvate when it moves into the mitochondria, it loses a CO2 and an NADH is made. We can then go around the cycle, more NADH is made, more NADH is made. An ATP is made here by substrate level phosphorylation. In other words, this reaction can't occur without ADP. We have an FADH made and another NADH made. Then we lose a CO2 here and a CO2 here, and did I highlight the third CO2? Am I missing it? Yeah, here, here, and here. 1, 2, 3. Okay? So 3 carbons enter and 3 CO2s come out. Now, if we look at the energy that is used or made, we get a delta G of a large minus number. In other words, we're producing energy. Where the real ATP or energy production occurs is what happens to these NADHs. So we have this 10 NADHs when this cycle turns for every 3 carbons. You get a tremendous amount of NADH and you get FADH too. Okay? And guess what? Where have you seen this before? Looks a lot like a chloroplast membrane, except it doesn't have photosystem 1 and photosystem 2. It has an entirely different set of electron carriers tied to, guess what, the ATP synthase. So what's happening in the mitochondria is the electrons that at one point were absorbed light energy now completely go through here and produce even more ATP. So NADH enters at complex 1, then 3 protons are produced, 4 protons are produced, 2 protons are produced. You have complex 1, complex 2, and complex 4, and you have ATP made. Got questions about that? Basically, you can see it's the same thing, except it's not using light to drive it. Now, this step right here, and let's follow the electrons through. So we're following the electrons through complex 1, complex 2, complex 3, and through the cytochrome into complex 4. And finally, we have this complex 4 here, and this is called the oxidase, the terminal oxidase. The electrons then reduce oxygen to water. Okay? Without oxygen, this entire system doesn't work. So as the oxygen levels decrease, this slows down. And this one step is why we need oxygen to survive. And it's why plants need oxygen to thrive and grow. If we competitively inhibit this reaction, or we block this reaction, we produce no ATP. And I don't know about plants, I assume it's about the same. In animals, we have maybe a 6-second supply of ATP. And so what's the number 1 molecule that you know that blocks this? Cyanide. Cyanide inhibits it. And very quickly inhibits it. So in animals, you inhibit ATP production, you die pretty quickly. Actually, cyanide does a lot of things. It binds the hemoglobin. So it kills you in several different ways. Okay? This step is why we need oxygen, why plants need oxygen. There's some more oxidases, and we use oxygen for other things, but it's minor. 99.999% of our oxygen is right here. So we take in oxygen, we give off CO2. Plants can do a lot of things. I think I was telling you last time, we can measure the amount of oxygen that we take up and the amount of CO2 we give off. It's called the respiratory quotient, and ours is always 1. We vary very little from 1. We just don't have any other things we can do. You know, if you put your head underwater for 5 minutes, your RQ is going to go to 0 because you're going to be dead. Okay, so in animals, it's almost always 1 O2 taken up for CO2 given off. In plants, it can be, the ratio can be all over, everywhere. It can be really high or really low. This is the electronic receptor. I'm getting ahead of myself. I'm not feeling very good today. And we can also do something with this. We can put plants in the presence of cyanide, and some plants, especially some seeds, will grow. So plants have an alternate oxidase, or an alternate pathway that's called cyanide-resistant respiration. So if we go here, here's why we need oxygen. Okay? And plants have evolved this other mechanism where NADH enters the chain and the electrons will go through this alternate oxidase where they use oxygen and, of course, water is produced. But what do you not see being produced in this pathway is ATP. Okay? So the alternate oxidase produces very little ATP. So why do plants have this? Well, one thing that you can see is that there are some plants that use this mechanism to actually heat up. Let's go back to this one. So if the rate of respiration, mitochondrial respiration goes down, the rate of glycolysis is going to go up to try to produce more ATP. Does that make sense? So in animals and in plants, you actually will heat up. So there are flowers that produce, I think one I usually talk about is, get ahead of myself, skunk cabbage. Yeah, and what happens when it starts flowering is the normal respiration is lowered. This alternate respiration increases, which then causes glycolysis to speed up and literally heat the plant up. And then that helps vaporize, you can imagine why it's called skunk cabbage. That helps vaporize the volatiles for pollinators. And I'm getting ahead of myself. The other thing is that it may be a mechanism to actually keep the plant from producing too much energy. Okay? So the thermogenesis one is something that people can actually say this is a reason. If this plant did not heat up, then it would not have evolved with that mechanism. It would not have survived. So we know that's why it does. The turn of the last century, there was a really popular diet aid that you could take that would cause you to burn more carbon through glycolysis because this diet aid inhibited mitochondrial respiration. Anybody know what it was? It was a really, really low concentration of cyanide. So long as you don't drink too much of it, you wouldn't die. And it wasn't on the market, as you can imagine, for very long. And it was, so you're uncoupling energy production. So you're gonna increase the only way you can produce energy is through glycolysis. You're gonna speed up glycolysis. It has a name for it. Anybody know what it's called? It's named after the guy that discovered it. It's called the Pasteur effect. So as long as you didn't take too much of it, you were fine. And then there was a period of time when people were eating apple seeds. Apple seeds have cyanide in them. And next time we're going to be talking about secondary metabolism. We're talking about a whole class of chemicals that are in plants that have cyanide attached to them. Okay? Questions about that? Okay, the last thing I want to talk about very quickly is something that's very important in seeds. And that is the breakdown of oils. And as you know, seeds store a lot of oil. What percent of corn oil? What percent of a corn seed is oil? Anybody know? It's less than 10%. But if you go to the supermarket, corn oil is one of the cheaper oils you can buy. Mainly because in the Midwest, in corn plants, corn goes in the front door and absolutely nothing comes out as a waste. 100% of the plant is used for something. And so when the seeds are used for oils, and then what's left is used for protein, meals, food source, animal feed. So even though corn oil doesn't have a lot of oil in it, it's one of the most common ones. Here's a list of some. So macadamia, 75% oil. That's a lot of oil. Maize, 5% here, so less than 10%. Sunflower, 40%. But again, there's just so much maize used. Palm oil is actually becoming popular for some reason. I think it's another fad diet thing that you should cook with palm oil in it. Anybody know anything about it? I've just seen it mentioned. So anyway, what goes on in the seeds? The seeds have an organelle that is very, very easy to see in a seed called a glycosome. And the glycosome and the mitochondria and the cytoplasm all keep this system running. The seed store oils as triglycerides, and they're hydrolyzed by lipases, and then the free fatty acids undergo beta oxidation, and then finally the acetyl-CoA that's produced starts what this cycle is called the glycosylate cycle. There are two enzymes that are unique in this cycle. One's isocitrate lyase and the other one's malate synthase. And these enzymes allow something to happen that can't happen in animals. That is, because of these enzymes, you don't need to know the pathways, you just need to know the glycosome, the mitochondria, and then finally, of course, the cytoplasm. Plants can take oil and turn the oil into sucrose without losing carbon. We can do this pathway, but we lose carbon. So plants can take a carbon here and put it into sucrose. So we have this very complex cycle, and we've actually been working on this for a while. And the products can go into the mitochondria, and then something that's interesting here happens. Glycolysis goes backwards. The enzymes in the glycolytic pathway are bidirectional, except for one. And so these PEP can enter and end up as producing sucrose, and then sucrose is going to be then transported in the embryo, into the embryo, or into wherever part of the plant's needed. Okay? It's called the glycosylate cycle. So all of these things, these pathways produce energy, and then they also produce all the carbon skeletons for everything the plant needs to make, which is everything. You know, plants need light, water, and minerals, and they make everything else. And we're going to finish up with this, which is what we started with. So we have this energy being produced in the pyruvate cycle, massive amounts of energy being produced, ATP, every time the glycosylate cycle, the TCA cycle turns, and then the byproducts then, or the products of these cycles, then feed out to produce everything else that the plant needs. So if we cut oxygen off, we shut down all of this. And the plant can survive for a good period of time just by producing ATP through the cycle here. Okay? Questions? Okay, I'm going to mention this very quickly, is that we do see changes in respiration over time. When we start talking about hormones, we'll be talking about ethylene, and you may be familiar with this burst in respiration that occurs when fruits ripen. Some fruits have it, some don't. And what we're looking at here is respiration. In respiration, we measure respiration as either oxygen uptake or CO2 being given off. And we see, of course, a rapid rate during growth because of the amount of energy that's required, not just the energy, but the substrates, the carbon skeletons. Then as the plant matures, it's in a maintenance phase, just like you are when you're sitting in front of the TV. You're not really moving or anything, so you're just making a minimal amount of what you need to make. And then in plants, we can also see this very rapid, sometimes incredible, and if you know about ethylene, this actually mirrors the increase in ethylene in plant tissue, which we'll talk about in a bit. Any questions? Okay, we're finished for today then. And I'll be here just a minute.